Okay, the second part is about brain before birth. And the question is how brain before birth would be affected by different environmental and genetic factors, and how the process of development for brain would be happening. We know that we start with just one simple egg, and then we have such a complex human being out of this simple egg. Not simple, just at least it seems simpler than the entire complex system of human being. But if we start from this simple egg, and then how this egg is moving forward in the process of childhood development or kind of fetal development to what we call from a simple egg to what we call blastola, and as we have here, it is called gastrola. So there is a typo here, so we need to have another R here. It's called gastrola because it's something like the stomach or the gastric cavity because of its shape. So we call it gastrola. So when we have this gastrola, there are steps in the process of brain development. I'm not going to discuss about those steps from the embryologic point of view, and there are some technical details and issues regarding the brain development. I will just mention few of those more important technical notes, and we will move forward with those notes to just have an idea about how brain would start to develop. The first issue is that when in the process of development, we get to a point that for fetus we have what we call a simple disc. So we have one simple disc that is supposed to make the kids. And in this disc we have different layers. So if I just make a cut from this, we have 3 layers. One outer layer, one middle layer, and one inside layer. So we call this ecto, meso, and endo. So and we call ectodermis, mesodermis, endodermis. And from this ecto part, which is supposed to make skin, we make basically a skin from the ectoderm. From this ectoderm, a piece from ectoderm would just go inside and eventually make what we call neural tube. Which is the building block of brain and spine. So this part of the ectoderm, which is a part of the future skin, would just go inside and make the neural tube. And that's the reason that we call it neurolation. Okay, that is the phase that we call neurolation. And neurolation would be happening here, and then this neural tube would evolve to what we call human brain and human spine, which is pretty much complex process. And it's not probably that much interesting for us right now in terms of the cognitive neuroscience, but few specific issues will be discussed today. So I will discuss about some of the technical issues that we have in this development. Let me just give you one of those issues in terms of, okay, as you can see here, in the first steps, it is just something like a one single tube. So as you can see here, it is just something like a one single tube across from the head side to the caudal side, to the kind of hind side. And this one single tube would eventually, over the process of time, would evolve to what we call human brain. So that is the process. And as here I can say that we call the ultimate steps of this process as what we call gyrification, as we discussed before, that when we have a newborn child, we have huge amount of gyri and sulci over the skull in this process of development from simply one single tube that is made from what we call ectodermis. So let me be back to the process. So we have started from this one neural tube, and then this neural tube developed the brain and the spine. And in this process, if we just want to see the process over time from sort of cellular level or cellular point of view, we are talking about things that are happening before birth. Okay, so we are in the prenatal phase. So we are talking about prenatal age. So in the 9 weeks after conception, we start to have what we call neurogenesis. So we start to have neural progenitor cells, or the cells are supposed to make neurons. And in this process, we also have what we call neural migration, differentiation, and maturation. And I will discuss about these steps along the discussion that we'll have over the time. But as you can see here, most of the neurons are basically being produced by the age of 18 weeks. And you can imagine any sort of insult to this system before the age of 18 would be able to change the amount or the numbers of neurons that we have inside the brain. And the process of neurogenesis would be kind of terminated almost during the time of the birth. In reality, we know right now neurogenesis would be happening for almost the entire life in some certain parts of the brain. So we know that neurogenesis would still be happening, but the extent of neurogenesis that we have in the kind of prenatal phase would be much more advanced and extensive around different parts of the brain. So we know that is called what we call neurogenesis, and then we will discuss about some other steps regarding the other processes regarding migration, differentiation, and maturation. So we'll discuss about those steps in the next slides. So that is the way that we see how the brain is being developed. As you can see here, when we have an early child after birth, we have the child with something like 300 grams of brain. So, and this is supposed to get to something like 1,400 grams. So we still have a significant amount of growth in the size of the brain, but the numbers of neurons that we receive by the time of birth would remain almost constant for the entire life. So we do not make huge amount of new neurons after birth. So most of the things are happening here before birth. Okay, so just having a sense about we receive all the neurons that we let's say 100 billion neurons that we have. We receive all of those 100 billion neurons at the time that we are born. Then we have the specific, really meticulous processes that would help in the process of brain development. Okay, one of these really interesting processes is called neural migration. So neural migration would be the process of having neurons being generated in an area that we call ventricular or periventricular zone inside the brain, and then migrating from these periventricular zones to what we call cortex and what we call cortical columns in the cortex. So they are being generated here, and there are what we call radial glial cell bodies that guide these neurons to just climb over these, as you can see here, climb these radial glial processes and go from the periventricular zone to the cortex. And this is a really interesting process of how these neurons would evolve and go to the cortex from some subcortical areas. And just to make it more simplified, this is the structure that we have in the brain. We have what we call ventricle, we have ventricular zone or periventricular zone, and we have white matter, and then we have cortex. As you can see here, these glial cells would just guide neurons to just go from the subcortical areas to the cortex and develop what we call cortex and what we call cortical columns. Okay, and there are specific prenatal insults that could make this process not happening in the proper way. And one of the things that could make this process with lots of difficulties would be different viral infections among pregnant moms, and that would make problem in terms of the immigration. And one of the most famous virus infections that could just basically prevent the immigration or migration process is called Zika virus. I'm sure that you have heard about that specific virus called Zika virus. And Zika virus is contributing to the kind of the inhibition of this process, or kind of making lots of hurdle in this process to at the end making lots of kind of early childhood developmental delays and complexities for brain development because of that viral infection. But that is the kind of the interesting process I told you, how these neurons would just migrate to the cortical areas and generate what we call all those 6 layers of cortical structures that we discussed about, different layers, if you remember, all those kind of beautifully connected different types of neurons would be generated in the subcortical areas, and then they just immigrate to the cortex. And based on the location that they are supposed to get settled inside, they just remain on the part that they are supposed to kind of get connected to other neurons. So they just go to their own site and remain there and get connected to other neurons to make the neural circuits that we'll discuss about them later on. The next process we discussed before is what we call gyrification, and that gyrification is a sort of a representation of all these neural migrations that are happening from subcortical areas to the cortical areas, and then we have an expansion in the space that we have in the cortical area, and this expansion would end up to gyrification, as you can see here inside the figure, that we have a great expansion in the cortical areas that would make cortical gyri and sulci and the evolved brain before birth. The third step in this process is what we call dendritic development and synoptogenesis. So we have neurons, but neurons should get connected to each other as well. The most important part is most of the neurons that we receive in a newborn child, they are not still connected to other neurons. And the process of connections and synoptogenesis would be happening after birth. So after birth, kids would start to just develop these synapses and make all those kind of beautiful connections together. As you can see here, even a 15 years old child would not have the level of sophistication and connections that we have in a 24 years old adult. So you can see that even from these steps, we are still in the process of making new connections and new formation of synapses. We will discuss about this in the next part of this class as well. But now we can get to the point that we have this issue in terms of, okay, what is the role of gene, or what are the things that genes would play significant role inside, and what is the role of environment? How environment contribute to the brain development, and how genes contributing to that development. And then we have the discussion regarding nature versus what we call nurture, and how they interact with each other through the process of interaction of genes and environment. To have a better sense about these concepts, let me just start to discuss about another issue that I'm sure that you are all aware about, and the process we call epigenetics. Epigenetics. And we know that when we talk epigenetics, is talking about, yes, we have genes that should be copied, and then they should be transcribed into mRNA and then being translated to proteins, and then their expression would be ending up to making phenotypes in human being or in different, let's say, neural cells. But we know that there are specific mechanisms like what we call DNA methylation that would be able to contribute to the process of having a gene off or having a gene on. So as you can see here, this gene is on right now. So the DNA is accessible here, but this gene is off here because of all these histone proteins that we have and the coils that these histones are making. This part of DNA is inaccessible and gene is inactive. Okay. The important part is there are some different factors that contribute in terms of how these genes would turn on or off, and one of these factors would be the factors that are related to our, let's say, personal experiences. So even there are ideas that if we experience trauma, the trauma could change these, let's say, DNA methylation processes or epigenetics factors in a way to make some genes off or on. The important part of this process is that these changes in gene expression could be transferred to the next generations as well. So that is the complexity of this process. So this is not about the genes that we have. It's also about whether these genes are turned off or turned on during these what we call epigenesis process. And this epigenesis process could be related to, let's say, as you can see here, development, environment, drugs, aging process, diet. I mean, all these factors could contribute to the expression levels of genes, and this expression level of genes could be transferred from one generation to the next generation to the next generation. And that is the complexity of this process. So the idea is if I receive a traumatic experience, that traumatic experience could be reflected to my genes in a way that kind of changes in my genetic material. It is not changing the actual gene codes, so we still have the same genetic code, but the way that these genes codes are being expressed and the pathway that they have for their expression would be affected in a way that change could be even transferred to the next generation, and then the next generation would be suffering from the consequences of the trauma that I have already received. And that is the complexity of what we are discussing right now. So there are already discussions in terms of, okay, if we have a minority in a population in the country, and that specific minority has experienced lots of social trauma or historical traumas over the time, how these historical traumas could be transferred from generation to generations to make a significant impact in the mental health of those who are living right now because of the traumatic experiences that they have experienced in their past generations. And that could be hundreds of years ago. So that is the complexity of what we have in terms of epigenetics. And it seems that epigenetics could contribute as an important factor in terms of how genetics could make an effect in terms of the development. And that, I mean, there are lots of discussions about the effect size and the items that could contribute to the epigenetics and the things that we can be done to redone some of the to redo some of the changes that we have in our epigenetics that could be affected by some environmental factors. So lots of discussion, interesting discussions, frustrations and positive things combined together. To have a better sense about how these interactions are interacting to each other, I might need to kind of mention another topic that is also an important topic for further discussions regarding the idea of what we call performationism, or epigenesis. Okay. Is there anybody here inside the class who would know what is the difference between epigenesis and performationism? And I should that epigenesis is not something related to, not very much to the term of epigenetics that I have mentioned just a few seconds ago. It goes back to Aristotle. So it is just a really kind of sort of old term that goes back to the time of something like 2300 years ago. So it goes back to the time that we had with Aristotle. Any comment about that? Anybody inside the class who would be able to have a sense about what is the difference between performationism? But that's a really hard term to express. Performationism and epigenesis. Okay, so let me just give you a sense about that, why they are different. In terms of the difference that we have, in performationism, what we are mentioning is that when we have child development, we have just a small baby in the sperm, and the small baby would just grow up in the female uterus and would make a complete human being. That is the idea that people had up to something like Middle Ages, and then even to something like up to something like 200-300 years ago. But obviously, you know, it is not something that we believe right now. We believe in what we call epigenesis, which is a formation of a human being or any other other creatures that we have from small building blocks that would kind of be produce and reproduce over time and making the entire being of these small components together. So that is the way that we call epigenesis compared to performationism, okay? But how epigenesis would be happening? So you just need, if you want to understand epigenesis, you just need to clear your mind from the epigenetics things that we have discussed. Epigenetics goes back to something like last 20-30 years ago. So it is a really new term. But what we call by epigenesis, it means that the genesis that would be related to kind of having different components putting together to make what we call the creation of a new creature in whether it's a plant or it's an animal or human being. And for what we call epigenesis, or the process of development from, I can just even say epigenesis to what we call the basic development from a cell to a complete system. Okay, so that development process is called epigenesis process. This epigenesis process could be what we call probabilistic or predetermined. And by predetermined, we mean that the process would be planned and controlled fully by genes. And by probabilistic, it means that based on the genes that we have, we can go to path 1, we can go to path 2, we can go to path 3, and probably not to path 4. So path 4 is not an option. But we have a range of different other pathways that we can go through, something like 10% here, 60% here, and 30% here. So we have different options. So, and there are probabilities for these options in terms of the gene compositions that we have. Okay, so we have different options. This one would be 0, something like that. And then, based on the environment, behavior that we make, and neural activations that we generate, that specific probabilistic genetics would contribute to individual development. Okay, so you might even go to the path of number 1 that was less probable because of the behaviors and environmental issues that you had around you. But there is still a role for gene. And it could be happening for things like, let's say, diabetes. So there are people who have this, let's say, path of something like 20% to get what we call diabetes mellitus before the age of 40 years old. So that is one path for some people. And They can get to that path, but it depends on the environment and behavior and neural activity, all the other things that they do to get this probability a reality or not. So that is what we call a probabilistic epigenetics. And this is how we believe right now the development process would be happening in terms of having genes providing us with different pathways, with different probabilities. So it is more probable to go to the path of number, let's say, here number 2, but you can also go to path number 1 as well if you do these sort of activities and behaviors, and if you live in a kind of this specific environment that would be environment that you make for yourself in terms of the physical activities you do, the type of culture that around you, or the type of social interaction that is happening around you. So this is how we consider, as we call, probabilistic epigenesis that would contribute to the process of development. And we know that this layer of interaction and contribution from all these 3 layers to the genetics, and then from genetics to the next generation, this is what we call epigenetics. And you can see here what I mean by genetics is different, but what we have been discussing about genesis. So genesis is about creation and development in general, and genetics is about, the science of genes. So just be careful about the way that you understand these terminologies. But one layer of these probabilistic epigenesis is what we call epigenetics, and how genes would contribute to this process. So I just try to make it more clear for you to understand how these processes are interacting with each other. And in this context, let me just give you a few examples as the last pieces of this part, and discussing about what we call fetal alcohol syndrome. So we know that if moms would drink alcohol in the process of brain development of their fetus or their unborn kids, we have what we call fetal alcohol syndrome in kids after their birth. And if you just evaluate their brain, kids that they had moms who have been drinking during the pregnancy, as you can see here, we are using a method that we call, if you remember, cortical thickness measurement, cortical thickness. Okay, and this cortical thickness, as you can see here, these areas, they have higher cortical thickness compared to the healthy control. And why higher cortical thickness would be important compared to healthy control, I will discuss about that in the next steps. And is it a good thing or bad thing, we'll discuss about that later on. So we'll discuss. So we know that their brain would be different. And fetal alcohol syndrome is not just about brain. Even in their faces, they have specific characteristics in their face that we call that specific facial expression of kids with fetal alcohol syndrome. So we'll discuss about those issues. Probably not here in cognitive neuroscience course, but just a sense about how environmental insults could make changes in the process of the brain development. And we all know about that, so we can kind of conclude, have a consensus that it is something that could make significant changes. Before ending up this part, I'm going to show you 2 slides regarding what we call the effect of marijuana use on prenatal brain development, and also the effect of prenatal smoking on brain. When you see these slides, the first slide and the second slide, if I just show you these figures in something like a scientific presentation talking about, okay, if you use pot, if you use cannabis, that cannabis use could make a significant impact on your kid's brain, and this is how your kid's brain would be on fire based on the figures that you can see here, especially the colors that we are using, the red color, so you can feel, okay, these are the parts of the brain that is being lost or being on fire based on the things that we can see here. Or if you smoke while you are pregnant, this is really bad, and this is the figure showing that how different parts of the brain are on fire because of your smoking in the kind of during the pregnancy. But in reality, this is what I'm expecting you as cognitive neuroscientists, based on what you have learned so far, to just make questions about these figures. When you see these figures, you need to know far more technical details to have an understanding and kind of be able to discuss about whether there is any effect for smoking or, let's say, marijuana use. I'm not going to recommend people to smoke or use cannabis during pregnancy. It is not, obviously, I'm not discussing about that. But it's also important for us as cognitive neuroscientists to have an understanding about, okay, what does it mean? I mean, what are these pictures are coming from, and what is the source of these images, and what do they really mean? And do they really mean what they are intended to show, or there are some technical details that we need to consider to have a better understanding about these figures. So please turn on your videos. I'm just going to ask few of those who are here. What are the questions that you need to answer to have an understanding about these figures if you want to, let's say, have any sort of conclusion about what do these figures are showing us in terms of the effect of prenatal cannabis or smoking in child development or child brain or brain development during prenatal age. Saina, could you ask one question about that? I mean, the one question that you are interested to know.
Actually, I need to think because I'm really new to this field.
Okay, you need to start to think about that. Okay. Ashkot, what is the question that you want to ask?
Nothing like pops up immediately, but maybe something interesting. Again, this is just, I haven't put a lot of thought in it. Maybe something interesting is that how that smoking, the way it impacts the brain, what is the connection from that section of the brain to the brain or the baby or the fetus that is growing? Is there any connection? Is there any, what should I say, neurological impact and connection between the brain of the mother and the child in the womb? I hope this makes sense.
Yeah, so you think that how mom, let's say, kid-brain interaction, let's say brain-brain interaction, would contribute to this process? One question that you're asking. What are the other things that people are interested to know when they want to have a sense about these figures? Mikhail, do you want to ask one question?
I think the first question should be, in what condition are these fMRI images taken? Because sometimes it's normal or even better to have hypoactivation or hyperactivation in certain areas. So what was the condition? What was the task? Is it a default mode, a resting state fMRI, or?
Michiel, let me just go back one step, kind of to the previous kind of state that we had. First of all, we are not sure that whether it is a structural MRI or fMRI. So that is the first question. I mean, if you are showing me this figure, the first thing I need to know whether these figures are structural MRI or functional MRI. So without having an understanding whether there are structural MRI or fMRI, I have no idea what do they mean. So that is one important factor. The second factor, as you have mentioned, if they are fMRI, what type of fMRI we are talking about? Okay, so let me, for example, saying that this one is a visual spatial task. Okay, so that is, it's a task-based fMRI using a visual spatial task, engaging your visual spatial cognition compared to, the control condition. So that is what we call the visual spatial task. So that is the second question that you need to answer. What are the other questions that you need to know to have an understanding of what do they mean? Puriya, not one other more question.
I'm going to ask, when was the fMRI or MRI taken? Was it when the child was still a child, or was the exposure in a childhood and now it's as an adult and we're checking the adult?
Exactly. So the third question would be, okay, how old are they? These figures. Let's say they are at the age of 18 years old. So we are talking about 18 years old sort of adults that they have moms that they reported that they have been using cannabis while they have been pregnant. Okay, so we are talking about brain images from those kids. So that is the third question. What would be the other question that you are interested to know? 4. Any others who are interested? I cannot read the names. Pariya, do you want to ask your question?
Can you hear me now?
Yes. Good.
I will ask myself that what functions are related to the affected parts, or what is the duty of each part, and what processes would be mainly affected by these failures?
What do you mean by process?
Which main brain processes are run by these parts, by these affected parts?
Okay, so the question that you are asking is, we have areas that they have, let's say, higher or lower activations, and you like to know whether these areas are contributing to specific functions or not. So that is the question that you have. Okay, good. That's a good question. What are the other questions that people are interested to ask?
I think another question could be, what was the condition of the mother? In what situation, emotional condition maybe, or which state of pregnancy, all those in condition of the subject.
So you are interested to know more about use conditions, right? So you really like to know, whether these moms, how many times they have used, have they reported just they think that they have ever used marijuana during pregnancy, or they have used more than, let's say, 10 times or more than 100 times, or they have been using frequently. So what is the intensity or severity of marijuana use when they have been pregnant? And what would be the duration, or when they have started, when they have stopped? So you need to have a better understanding about the intensity. So we are talking about heavy cannabis users, or we are talking about somebody who has tried once cannabis while they have been pregnant. So what are the technical details we know, and obviously how much we can rely on the memories that are related to something like 20 years ago. So that is one other condition that you might be asking about. What is the exposure, more technical details about the exposure that these kids received. What are the other questions?
And I was more trying to mention what are the other symptoms that this person has? Because if you want to compare, for example, did she have any depression? Was she running frequently, have any exercise? Because those things could actually incorporate to the result. That was what was kind of in my mind.
Of course. And so let's say I should add another item 5. comorbidities. Those who have, those moms, have they been experiencing any mental health issues? Have they had any sort of, let's say, traumas on that period of time in their life? I mean, you can imagine a mom who is still using cannabis every day in pregnancy. You can imagine the social context, the other complexities around them, how you are controlling those factors, how much you are pretty much sure about the challenges that are related to those, let's say, other contextual factors and comorbidities that are associated with cannabis use, and how much you're controlling them, being able to say that, okay, this is the effect of marijuana use during pregnancy on children's brain. So that is another complexity. What else do you want to ask? Let me see, I can see Clemence. Clemence, do you want to ask your question?
Yes, me with my experience, when I'm looking at that, I would need the coordinates of the regions we are looking at.
Okay, how coordinates are going to help you?
Because I don't know if it's more lateral or really in the middle, especially for the subcortical regions. I think for an experienced doctor, it might be more easier, but when I'm looking at that, I really don't know what I'm looking at.
Okay, I do agree. So you say that the figure is not enough to give you detailed information in terms of, okay, what are they really located? So the figure is just giving me some blobs that might be hard for me to completely define where are they exactly located. So I do agree. So you probably need to have more than just figure. You need also to have tables, you need to have maps, you need to have more technical details. So this is not enough for you as a cognitive neuroscientist to know what are the areas that are affected. So I do agree. So you need to have more technical details. That's fair. So the idea is having two groups of kids. One group of kids that they have parents that they reported they have been using marijuana. I mean, they have moms that they have been using marijuana when they have been pregnant. And another group of kids that they have, let's say, moms that they have reported that they have never used marijuana in their life. So let's say you go to a school, you have 200 kids in the school, you just do the scanning on those kids, and then you also ask their moms whether they have been using cannabis or not, and then you divide them in two groups. One group, their mom reported that they have been using cannabis. The other group, they report they have not been using cannabis, and you compare them in an fMRI task regarding visuospatial cognition, and you show that one group has higher activation in some areas compared to the other group. And you say that, okay, this is the effect of prenatal marijuana use, and this shows that the brain is seriously affected by marijuana use, and you start to recommend people you shouldn't use marijuana because of this figure I'm showing you.
So we can directly conclude this. I mean, 18-year-old is too much, and the other conditions the child might have had during childhood, or
yeah, even in reality, as Mikhail has mentioned in the first part of the discussion, we really don't know whether higher activation or lower activation is a better thing or a worse thing. If people have higher activation in some areas, it's really hard to say whether they have a better performance or not, especially when they report that there is no significant difference in terms of their behavior for that task. So the function of the task is the same, but one group has higher activation in one area, some areas, and some deactivations or lower activation on the other areas, and there are significant differences between groups. And using that as a tool to show to the public audience that there is a significant negative effect of cannabis use in pregnancy for kids, and you should avoid doing that. So there are, I'm not going to, probably I can discuss about this for an entire 2-hour session, discussing about the technical details that we need to consider when we are using these beautiful brain images with the specific blobs of red dots, and just trying to use them as a way to justify doing something. For example, Ashkan is interested to, let's say, doing yoga, so he starts to just bringing kind of kids with two groups of with moms who have been doing yoga during pregnancy and the other group who have not been doing yoga during pregnancy, and trying to show, okay, now at the age of 15 years old, those kids have different brain activations. And that is the reason that I highly recommend all moms to do yoga, because I mean, these kids with, I mean, yoga, I mean, there is a significant difference between these brains at the age of 15 years old because of their moms have been doing yoga when they have been pregnant with those kids. So there are, I was just trying to say that these beautiful pictures, I mean, whether this is the picture we are discussing or this is another picture that we have, they are really, I mean, it's really important for us to have a, first of all, have a really good understanding about these figures. We need to clearly know what they are talking about, what is the specific task that we are talking about, how these groups are being recruited, what are the contextual kind of information that we need to have, how these two groups are being matched for their socioeconomic status, different environmental factors, and how there could be an effect that could be detected by fMRI, and is there any meaningful effect? We can just have, let's say, two groups of kids. One group is more anxious because of the social context that they have when they go inside the fMRI scanner, and just because of having a higher level of anxiety when they are doing fMRI tasks compared to other group of kids that they have lower level of anxiety, all because of their social context, they can have different activations at the end of the day. But when you go back to the changes, you can see just it is just because of some, let's say, contextual factors that would run in the type A family compared to type B family. And then you have different levels of activation inside the scanner, and it is not related to any sort of brain developmental change. So I'm not, at the end of the day, I'm not saying that please go ahead and use kind of cannabis while you are pregnant. It is not about that. It's just about how we are using the brain imaging methods to justify things. So we need to be really careful in terms of kind of how we use these pieces of evidence. And of course, when something is being published in a prestigious journal, definitely reviewers ask about those things, and they have tried to make sure that all these considerations are taking or are kind of supposed to be kind of taken into account or taken into considerations. But I realize that there are, especially when you talk about publications that goes back something like 10 years ago, and they have been the first publications in the field, and sometimes you can have reviewers that, I mean, they really like to show that there is an effect because of kind of during pregnancy drug use, and they want to encourage others not to use that, so they lower the level of threshold to accept it. So I just wanted to mention that you need to be really careful in terms of how to read these publications. And I highly recommend you, when you want to kind of read about the materials that we have for this class, and just going to the publications and see the actual paper and go into the details and see how they have tried to match. So this is what I'm expecting you to do as a kind of graduate of this cognitive neuroscience course, to critically think about the figures that you see and just not to be amazed by the beauty of these brainish pieces of evidence that we have, and these beautiful, let's say, blobs of activations or changes in the structure that we can show, and just amaze people with these kind of findings that we have from the brain without having a good understanding about how we are making these pieces of evidence.